Hello students, welcome to the second lecture of the first chapter, Herbs as Raw Material, of the subject Herbal Drug Technology BP603T. I'm Dr. Kamal Madan, Associate Professor, Lloyd Institute of Management and Technology. Now we need to study the topic called Selection of Herbs. We have already done what are herbs, where do we get from, what are the various sources. Now we need to select those herbs so that we can process it and we can go for the industrialization. Selection of herb involve the species or the botanical variety that should be the same as specified in the official pharmacopoeia. We know the pharmacopoeia. It is an official book or the literature, or you can also call it as ग्रंथ. तो जिस तरीके से ऑफिशियल फार्माकोपिया में स्पेसिफिकेशंस दिया हुआ है प्लांट का, सिमिलरली दैट इज़ द वन विच इज़ दैट ऑफ द ऑथेंटिक सोर्स. अगर आप भी कोई प्लांट पार्ट कलेक्ट करते हैं या सेलेक्ट करते हैं कलेक्शन के लिए या रिसर्च के लिए, सो यू नीड टू ऑथेंटिकेट द सेम. सो वे यू विल गेट ऑथेंटिकेटेड from that official pharmacopoeia characteristics which are mentioned there. The plant variety selected for cultivation should be identified and documented. आप जिस भी सोर्स से कलेक्ट कर रहे हैं, जहाँ से भी कर रहे हैं, आपको उसको अच्छे से आइडेंटिफाई करना है. You should be sure of this is the same genus, same species, same variety, same family. Everything you have to first make sure and only then you have to collect it. So for that what you need to do is आप उसको अच्छे से आइडेंटिफाई करें और फिर उसको डॉक्यूमेंट करें. Identification and authentication. Now, if we talk about the botanical identity, the species, subspecies, genus, variety of the plant for cultivation should be verified from a qualified botanist or institute and they are recorded. Whenever you collect a plant, you get it authenticated from a botanist. Normally, universities में botany department में जो भी डील करते हैं इससे, you have to show them the plant part or the plant and they will then going to authenticate. वो आपको श्योर करेंगे कि ये वही जीनस है, वही species है, because they have the herbarium maintained with them from where they generally identify. Or specimens. A specimen of the plant should be submitted to the National Herbarium for identification and documented. And seed and other propagated material, how they are identified. The suppliers of the seeds and other propagation material should specify all the necessary information related to their identity, quality, as well as their breeding history. So these are the different ways by which you identify the herb. What is the importance of identification? It is very much necessary. You may be confused between two different varieties. They are having similar leaves, they are having similar stem length, but they may be differing in the active constituents. So identification and authentication is a foremost step to ensure the purity as well as the quality of the herb. 90% of the crude drugs are obtained from the different plant sources and just 10% are obtained from the animal and mineral sources. Specification for herbal starting materials are intended for quality, as I said you earlier. Characteristics are found to be useful in ensuring safety and efficacy. Quality of herb depends upon the environmental conditions under which it is grown. You might have seen the differences in the feature of a plant or a fruit which is grown in a different condition. आप देखिए, say for example, you eat bear, right? So one which exists in the North India, the one which exists in the South India, they differ. Why do they differ? Because of the different weather condition, different environmental conditions. So they give the different characteristics to the plant. इसीलिए प्लांट की आइडेंटिफिकेशन और ऑथेंटिकेशन बहुत इम्पॉर्टेंट है. The characterization is also essential for the establishment of the specification. Botanical और phytochemical aspects से भी आपको उसकी specification को establish करना ज़रूरी है. What are the factors for identifying? जब आप किसी प्लांट को आइडेंटिफाई करें कि ये वही प्लांट है जो आपको चाहिए ही कि नहीं है, तो आपको कौन कौन से फैक्टर्स, कौन कौन से पॉइंट्स को ध्यान में रखना होगा, राइट? द ड्रग्स ऑफ द प्लांट और ओरिजिन, होल प्लांट, आपको पूरा होल प्लांट के सारे फीचर्स कंसिडर करने हैं या नहीं करने हैं, या उसके किसी पार्ट के करने हैं, या फिर उसकी रूट के करने हैं, स्टेम, लीफ, फ्लावर, सीड, fruit modification of the stem and root, bark of a stem or root or wood or their exudate or gum. So you have to identify exactly which plant part or which whole plant is your herb which you have to target, which you have to select. Other factors which you need to consider are surface size and surface shape. Shape कैसा है, oblong है, oval है, whatever, you have to identify that. Sensory और organoleptic characteristics, इसमें आते हैं आपके color. What is the color which you need to identify? आपको red color के flowers लेने हैं कि pink color के लेने हैं कि orange color के लेने हैं? What is the odor? How does it smell? You have to identify those organoleptic properties also. How does it taste? What is the consistency? इसके अलावा विजुअल कैरेक्टरिस्टिक्स भी एक फैक्टर है जिससे आप प्लांट को आइडेंटिफाई कर सकते हैं. यू हैव हियर द साइज, कलर, सरफेस कैरेक्टरिस्टिक्स, टेक्सचर एंड फ्रैक्चर कैरेक्टरिस्टिक्स. जब आप उसको फ्रैक्चर करते हैं, प्लांट की रूट को या स्टेम को एकदम से तोड़ते हैं, तो कैसी आवाज़ आती है? स्नैपशॉट आता है, या शार्प आती है, या ब्रिटल आता है. सो दैट इज़ ऑल्सो वन ऑफ द आइडेंटिकल फीचर्स, दैट इज़ द फ्रैक्चर कैरेक्टरिस्टिक्स. Now, when you need to identify the plant, what you need to identify exactly? उसकी botanical name, कौन सी genus को वो belong करता है, क्या उसकी species हैं, क्या सब species हैं, या क्या further variety है, what family it belongs to, what is the source, like it is one, say केसर जो इंडिया में है या केसर जो ईरान में है, right? What is the source of that plant? Because again, depending upon the different environmental condition and the different sources, they have different features. What is the origin? Originally वो plant कहाँ था? Because the moment it transfers from one place to another, from one geographical location to another, the features also changes. What about the description? Macroscopically वो कैसा दिखता है? Size कैसा है? Shape कैसा है? Color कैसा है? Surface कैसा है? These come under the macroscopic features. And the microscopic characteristics which tells what are the types of the cells. Guard cells कैसे हैं? Stomata ऊपर है कि नीचे है? Epidermal layer कैसी है? So that all comes under factors for the identification. Then besides these factors, you have some tests which help you in the identification. Now these tests for identification include TLC, that is your thin layer chromatography, TLC. Thin layer chromatography is the chromatography or the analytical technique जहाँ पे आप एक pure compound को identify करते हैं. This technique is normally carried out in the labs also. Students of the undergraduate also perform it. You basically have a silica plate. आप एक silica plate prepare करते हो, आप उस पर एक mixture of अपना जो भी solvents का है उसको apply करते हो. You have the mixture of the active constituents, and when you want to identify a particular phytoconstituent or particular active constituent, you put a mark here. Now you basically see how much distance does it travel. So that is the RF value which helps in the fingerprinting to identify that particular active constituent. Then assay. Assay is what? Assay is basically a quantitative analysis. आप ऐसे से आइडेंटिफाई करते हैं कि उस प्लांट पार्ट के अंदर कौन सा कंपोनेंट कितनी अमाउंट में है. ऐसे का प्रोसीजर प्लांट के ऑफिशियल फार्माकोपिया लिटरेचर से आपको मिलता है. So for that you also need the marker. Marker means the pure compound. Say for example, if I want to go for the अजवाइन leaves, अजवाइन आप सबने घर में देखा होगा. Now this अजवाइन contains a terpene called thymol. अब इस अजवाइन की अगर मैं एक्सट्रैक्शन करती हूँ तो इसके अंदर वो सारे कंपोनेंट्स आएंगे जो इसके अंदर हैं, लेकिन मुझे किस पे फोकस करना है? आई हैव टू फोकस से ऑन थाइमोल. सो व्हाट डू आई नीड टू डू इज आई हैव टू हैव अ प्योर सैम्पल ऑफ थाइमोल ऑल्सो, सो दैट मैं आइडेंटिफाई कर पाऊँ कि मेरे मिक्सचर में कितना थाइमोल है एज कंपेयर टू दी प्योर थाइमोल. सो उस प्योर थाइमोल को हम कहते हैं मार्कर, राइट? Now, the other factors are test. You have to carry out the test for the toxic material, pesticides and the microbial contamination. So, these are the different factors which a botanist, which a herb scientist or which a pharmacognosist need to keep in the mind before the identification of the plant. Herbarium. Now, what is herbarium? आप लोगों ने स्कूल लेवल पे भी एंड कॉलेज लेवल पे भी एक हर्बेरियम फाइल ज़रूर बनाई होगी. You may not be able to focus, but do you remember we used to collect the leaves of the different plant parts. We used to dry those leaves and we used to preserve in a file. आप शीट में लीफ लगाते थे, उसको लेबल करते थे, सबमिट करते थे. That was just one simple most herbarium. So what is herbarium actually? It is an important scientific record for the flora. Flora means plants. It is containing the pressed collection or depository of the dried plant specimen. हम plant के part को dry करते हैं. Dry क्यों करते हैं? Because अगर moisture रहेगा तो moisture is going to lead to the microbial contamination. उसके पर fungus grow कर सकता है, right? Then this herbarium displays part of each plant which is labeled with the scientific and the common name. It also contains description of the species, form, habitat and location. It is an accepted system of classification for future reference of study. इसका basically purpose क्या है? When in future you're going to collect the same plant part, so you can refer to the herbarium and you can establish its identity. Say for example, you have these sheets with you. Now I have dried a leaf and pasted this leaf, and here I am going to give the all details. I am going to give all the details, all the information about this leaf. Now this information that has to be its plant genus name, its species, its habitat, its location, आपने कहाँ से collect किया, कौन सी species है, what is the environment you have taken care of from, exactly आपने कहाँ से collect किया. So that becomes your herbarium, and whenever you need to collect the plant, the same plant, then you can refer to the herbarium so that it helps you in the reference and the study, right? Now, here comes the herbarium specimen label I was talking about. तो जब भी herbarium फाइल बनाई जाती है या herbarium बनाया जाता है, उसके अंदर कौन कौन सी information रहती है, what data does it contain? It contains the scientific name. Scientific name कहें तो genus followed by species like Solanum tuberosum, and yes, if there is a particular variety as well, and the family it belongs to. डिटेल्ड लोकेशन, कहाँ से वो प्लांट का पार्ट या वो प्लांट लिया गया है. ऑरिजिन, वेयर डज इट एक्चुअली एग्जिस्ट. हैबिटेट, वेदर इट वॉज एग्जिस्टिंग इन सॉइल विच वॉज मॉइस्ट और ड्राई सॉइल, किस हैबिटेट में वो प्लांट पार्ट था जब आपने लिया था. डिस्क्रिप्शन, अगेन हाउ डज इट लुक, उसके सारे मॉर्फोलॉजिकल फीचर्स, what are the shape of the leaves, what are the color of the leaves, what is the surface of the leaf, does it has any odor, कुछ अलग से उसकी smell है जिससे आप उसको identify कर पा रहे हैं. Then collector name, जिसने भी वो plant part collect किया है उस particular habitat से. किस दिन कलेक्ट किया है, you have to put the date of collection. But yes, why does it make a difference? Because during the stage of a plant growth, the bioactive constituents, they vary. Say for example, belladonna की लीफ का हम तभी कलेक्शन करते हैं जब उसकी फ्लावरिंग का टाइम होता है. क्यों? क्योंकि तब उसके अंदर मैक्सिमम अल्कोलॉइड्स होते हैं. So you have to make sure at what time of the year are you collecting it, or at what stage of its growth you are going to collect it. So you have to mention the date of collection and the project information, why you have been doing this herbarium specimen label actually. So proceeding with the herb authentication, yes, how do we basically authenticate the herb? We authenticate the herb on the basis of its botanical or the pharmacognostic characteristics. We have to see that whatever commercial variety we are taking, that does not be having any sort of substitution or any kind of adulteration. All this we are able to achieve only when we are able to establish its pharmacognostic characteristics. Whatever the features it consists, say its macroscopical feature. If we talk about the leaves, leaves का color कैसा है, leaves का size कैसा है, leaf का surface कैसा है, does it have some latex coating over it or not? Or if we are talking about the flowers, flowers के कितने sepals हैं, कितने petals हैं, what is the structure of its stamen, what else does it contain? Then we have to look for its microscopical characteristics. हमें आइडेंटिफाई करना है कि उसकी जो सेल असेंबली है, उसकी जो टिश्यूज़ हैं, उसका ऑर्गेनाइजेशन कैसा है, राइट. देन वी हैव टू ऑल्सो लुक फॉर इट से मॉइस्चर कंटेंट कितना है. इफ़ यू आर टेकिंग द सीड पाउडर, तो उस सीड पाउडर की वाटर एब्जॉर्प्शन कैपेसिटी कितनी है, पाउडर के कैरेक्टरिस्टिक्स क्या हैं, what are the extractive it contains. if we talking about say water, अगर solvent यूज़ कर रहे हैं तो उसका water extractive कितना है, या फिर उसका इथर एक्सट्रैक्टिव कितना है, या एसीटोन एक्सट्रैक्टिव कितना है, या पेट्रोलियम ईथर एक्सट्रैक्टिव कितना है. सो वी हैव टू लुक फॉर से लॉस ऑन ड्राइंग. ये सारे के सारे जो भी फार्माकोग्नोस्टिक टेस्ट हैं, पैरामीटर्स हैं, दे आर ऑल गिवन इन द फार्माकोपिया. From there you can have the procedure, how do you basically perform that, what is the content of the active constituent contain, क्या उसके अंदर alkaloids हैं, क्या उसके अंदर tannins हैं, अगर हैं तो कितने हैं. You have so many tests accordingly which you need to perform in order to authenticate the herb. So that is called as pharmacognostic characteristic. Now what do we do here is the method of authentication of the raw material, who is doing this and for what purpose, describes how the original material or the plant was identified and allows the reader to determine to some degree if raw material was produced from the plant as reported. जब ये सारे फीचर्स आपको ऑथेंटिकेटेड सोर्स के अंदर मेंशन होंगे तो कोई भी फर्दर रीडर उनको मेंशन कर सकता है, उनको एड्रेस कर सकता है, उनको स्टडी कर सकता है, और फ्यूचर अपनी रिसर्च के लिए अगर दोबारा से उसी प्लांट को यूज़ करना है तो उसको ऑथेंटिकेशन को, उसकी प्लांट की आइडेंटिफिकेशन को इस्टेब्लिश कर सकता है. So that herb authentication that is being assigned as a lot number. Then after establishing its identification, it is given a lot number, a number which going to tell its quality establishment of the raw material, provide the reader with the key information as to where from the raw material came. So this is the herb identification. Now the herb identification या फिर herb authentication जो process है, this is done by the, on a smaller scale, as I told you earlier, it is done by any botanist of botany department in the university, fine. Or the other way is there are large number of the bodies which are involved with this. So this is your National Medicinal Plant Board, NMPB. This comes under the Ministry of Ayush and it is situated in Delhi. Then you have National Institute of Science, Communication and Research, which basically comes under CSIR. CSIR is Council of Scientific and Industrial Research. This is the largest research and development organization of our country, and the president of this organization is the Prime Minister of that country. Then we have CIMAP, which is Council of Medicinal and Aromatic Plant, and this is situated in the Lucknow. So this is a frontier organization which deal with the research in the medicinal and the aromatic plants. So this CIMAP also come under the CSIR, and this NISCARE also come under the CSIR. So this CSIR basically the largest R&D organization which is an autonomous body under the government of India, which deals with the authentication of the plants and their herbs and as well as their parts. So that's all for the lecture today, students, and I shall be taking up in the next lecture. See you again.